Okay, so in this one we're going to talk about how the immune system deals with bacterial pathogens. We're going to go in depth on how it deals with specifically extracellular and intracellular bacteria, because obviously there's different responses. And we're also going to go a bit into how the immune system deals with fungi. So let's just get into it. The first lines of defense against bacterial infection are the epithelia, the microbial peptides on our cell surfaces, so our antimicrobial factors, which we talked about when we were discussing the microbiome, and also the mucous membranes, because these catch bacteria and our cilia on top of our epithelial cells fan bacteria away from these mucosal sites that are gates of entry for potential pathogens. So immune cells then phagocytize pathogens, and antibodies and complements opsonize them to make them easier for phagocytes to actually envelop and destroy. We also have acute phase proteins that repair tissue and kill microbes. So these are some of the kind of the first lines of defense against these bacteria. So think of it as kind of like an outer wall, defending the body from defending the body from these bacteria actually getting in and starting the infections in the first place. And then we have our innate and adaptive immune responses with our T cells and B cells. So with regards to extracellular bacteria, bacteria that proliferate outside of host cells, CD4 T cells play a role in defense against these bacterial infections by producing cytokines that attract B cells, which will then produce antibodies to opsonize these extracellular bacteria, by producing interferon gamma to activate macrophages that will phagocytize these extracellular bacteria, and by producing TNF to trigger an inflammatory response, which will fill the site of infection with lymph, as well as other cells from the lymphatic system that we need to deal with these infections. Bacterial antigens tend to produce a T cell dependent response within B cells. So we talked about that when we talked about immunoglobulin isotype switching, where a naive mature B cell is going to receive signals from a follicular T cell, causing isotype switching to go from producing ITA or ITM to producing the immunoglobulin isotype immunoglobulin G. And IgG opsonizes proteins quickly and with high affinity, as well as activating complements. These complements can either opsonize the extracellular bacteria, or they can encourage a process called chemotaxis, which is the official word that we use in immunology to describe the movement of leukocytes to a site of infection. Leukocytes just referring to kind of white blood cells, cells of your innate immune system, like our macrophages, our neutrophils, our monocytes, just recruiting them to the site of infection. The proper term is called chemotaxis. And recall from when we talked about the different types of T cells, the different types of helper T cells and their functions, that Th2 and Th17 specifically deal with extracellular pathogens like these extracellular bacteria. In contrast, for intracellular bacteria, an example of which being mycobacteria, which are responsible for tuberculosis and leprosy, these mycobacteria proliferate within host cells, within structures called phagosomes, and they don't need to worry about being opsonized by antibodies produced by B cells because they're dividing within host cells, so they're not going to have to worry about that or being phagocytized outside of a host cell. Cell-mediated immunity is extremely important when it comes to dealing with these infections because granuloma formation by T cells is required to make these intracellular bacteria dormant. So the main example being granuloma formation within the lungs to put tuberculosis into its dormant state. If the granuloma contains more Th1, then the tuberculosis bacilli will be contained within the granuloma, which is just a structure with connective tissues. It can be calcified, surrounded with kind of macrophages, monocytes, neutrophils, and a lot of these helper T cells. So if it's more Th1, then the TB is going to be contained within the granuloma, because the Th1 specifically deal with intracellular pathogens and produce cytokines that can deal with them. Whereas if the granuloma actually contains more of these Th2, which are more designed to deal with extracellular bacteria, then they're not going to be producing the right cytokines. So the tuberculosis bacilli will not be contained within a granuloma, and you'll have active TB, which will spread throughout the lungs and infect more macrophages. Bacteria also have methods of evading the adaptive immune system. So bacteria may inhibit chemotaxis to prevent immune cells being recruited to the site of infection, like we talked about a moment ago. Bacteria are also capable of destroying antibodies with hydrolytic enzymes, as well as altering antigens via bacterial genetic recombination to give their antigens different epitopes, so the host needs to trigger a new adaptive immune response to make complementary immunoglobulins and T cells. Bacteria are also able to release toxins capable of killing phagocytes, like macrophages, so if they're killing them, they're not able to infect them. The macrophages, the phagocytes aren't able to kill the bacteria because they're producing toxins that kill them first. Mycobacteria, like we talked about a moment ago, have unique strategies of invading immune cells. For instance, they force macrophages to produce immunosuppressive cytokines. They dwell within the macrophage itself, so they avoid being killed by the phagolysosome of the macrophage. They inhibit the phagolysosome, and they also induce production of interleukin-4 and interleukin-10 within the macrophage, which are anti-inflammatory cytokines, so we're not going to get the inflammatory response that is going to recruit other T cells and B cells from the lymphatic system to the site of infection, as well as other stuff like neutrophils and lymph that we need. They're also capable of inhibiting antigen loading on MHC, so the MHC class 1 and 2 aren't able to present antigens to helper T cells or cytotoxic T cells to produce a B cell response or to induce apoptosis of internally infected cells. And they're also capable of inhibiting host lipid pathways by modulating the kinds of leukotrienes and other inflammatory mediators produced by the macrophage in the granuloma. So finishing off quickly, we'll go over fungi next time. Some bacteria, like Staphylococcus, are capable of producing things called superantigens, like enterotoxins, which basically binds to the MHC class 1 of antigen-presenting cells, and they're recognized by T cell receptor on helper T cells outside of the antigen binding site, which results in activation of antigen non-specific T cells, which induce other inflammatory responses by secreting cytokines like TNF alpha, TNF gamma, interleukin 17, interleukin 16. We'll go over it